Hello, class. I hope you're all doing well. Chapter 22. It's about DNA. I brought my son's toy, so I will, I was able to steal it from him. And then, so you will see this a lot. And then, how does DNA, from DNA, we get to protein molecule. See, this is a lot of me. Look at my hair, my skin color, my eyes, my teeth. Pretty much we are made up of protein. And based on this blueprint, so on a piece of paper, for example, there's a blueprint of a building, right? They draw it on a paper first, and then the building goes up. Same thing as far as a cell goes. Inside of the cell, there is DNA, and based on this DNA, this goes up. So I hope you got the big picture of this chapter. And then toward the end of the chapter, they talk about your textbook talks about a few things that recently we human are able to manipulate. We can manipulate this and have a hopefully a better lifestyle and better products and so on and so forth. So we'll talk about that. So first, let's talk about the structure of DNA. As you know, DNA is a deoxyribose. Where is my pen? Deoxy, it means right here, deoxy, it means without oxygen. Without oxygen. So this is a nitrogenous base. You will see that the sugar molecule, which is attached to the nitrogenous bases, we will see here in a minute. They do not have, the sugar molecule does not have the ribose, if you would. The sugar molecule is called ribose. It does not have oxygen. On the other hand, RNA, you will see it does have oxygen. I think we talked about these differences at the beginning of the semester, chapter 3, I believe. Anyhow, so a gene already gave you this definition of a gene from beginning of semester until now. So it's just a piece of DNA, which is part of a chromosome. Imagine my arm is chromosome. My arm is made up of many of these ladder things. Okay, so this would be a gene, a segment of traits. So genes are organized into chromosomes wrapped around the protein molecule called histone. I don't want to bore you with a lot of extra information. So histone are the protein molecules that DNA, imagine this is DNA, it wraps around my fist is histone, it wraps around that I told you. I cannot fold my ladder, but if this ladder folds around my arm twice and extend to another fist, so these fists are called histone protein, protein. Located, of course, DNA is located in eukaryotic cells. They are inside of nucleus. In like bacteria, they are in a nucleoid area. They do not have nuclear membrane, bacteria, and all of those that we talked about it. And of course, there is DNA found in mitochondria and also in chloroplast. So plants have chloroplast, they do photosynthesis with the help of chloroplast, and then you have them in chloroplast as well. Deoxyribonucleic three main functions: replicate, so it can be transmitted to the next generation. Of course, it has to be replicated. Stores information, DNA information for making protein synthesis. Undergoes changes. It happens, mutation to provide genetic variability. DNA is double helix. So this ladder is double, one leg. That's what they mean, double. That's one means. And helix, I cannot do it with this. But when it's twisted like this, when it's twisted, it means it's helix. So DNA is twisted like this. So that you guys can see that in my diagram. That's what double helix means. Okay. So composed of two strands, I already talked about that. And each strand is a series of nucleotides, which I will show you that. Nucleotides are made up of three subunits, a phosphate, a pentose, which is deoxyribose. It does not, the sugar molecule pentose, it has 5 sides. And it does not have one of the carbons attached to it. Its hydrogen is not OH, hydroxyl group. And a nitrogenous base containing base, which are adenine, cytosine, guanine, thymine. DNA has only those 4: adenine, cytosine, guanine, thymine. Adenine and guanine are purines. Purines, it means they have 2 rings, okay? And then cytosine and thymine, they are pyrimidine. They are 1 ring, okay? So adenine and guanine, they have 2 rings, purines, and then cytosine and guanine, they're pyrimidine. Right here, so you can look at it. So these are the sugar molecules right here, the deoxyribose. Here is a phosphate. So the leg of the ladder are made up of phosphate would be right here, sugar, phosphate, sugar, sugar, sorry about that. Sugar, well, right here, sugar, phosphate, sugar, phosphate, sugar, phosphate. Same as this side. Sugar, phosphate. You guys see that? Maybe I should bring it closer. So this would be a phosphate, sugar, phosphate, sugar, phosphate, sugar. I hope you guys can see that now. So that's what this is. The legs of the ladder are made up of sugar, phosphate, sugar, phosphate, sugar, phosphate. The steps of the ladder on this side is a purine, on that side is a pyrimidine. Purine, pyrimidine. Well, if it is A, it is T on the other side. Adenine on this side, T on the other side. So it's always A and T bind together, okay? And then G and C bind together. What are these 2 lines? Why this one has 2 lines and this one has 3 lines, Amir? Well, because there are 3 hydrogen bonds between guanine and cytosine. There are only 2 hydrogen bonds between adenine and thymine. Okay? And the rest, that's how it is. We are made up of, all of living organisms on planet Earth, everything. I'm not including viruses, bacteria, and all the way up to elephants, whales. We are made up of 4 letters. Believe it or not. We are made up of adenine, thymine, guanine, cytosine. The blueprint for all of us is made up of these 4 letters. In computers, it's 0, 1, right? The programming, those of you who are into computers, 0, 1. Well, 4, that's it. Believe it or not. Okay, so that's a DNA that helix, sometimes they call it the alpha helix, the stereotoroid. So it is a double helix. That's the structure of, and these are the legs of the rungs, the legs of the ladder. These are the rungs of the ladder. But anyhow, let's move on. The phosphate and sugar molecules make up the backbone of the project, and then bases are joined by hydrogen bonds, which is called complementary bases. So it's called law of base pairing. This is called law of base pairing. Okay, so A and T must bound together. Okay, and G and C must bond together. 3 hydrogen bonds between them. Here they are showing it to you. And then it's the structure of DNA. You might see, this is, you guys pay attention to this. This is upside down, right? They draw this upside down. So it's, this structure is anti-parallel. Okay, so it's anti-parallel. DNA, these 2 legs of ladder, if you look at them, then you're seeing it here right now. They are not parallel to each other. They're anti-parallel. One goes down and the other one is going up. Okay, so, and here we go. And the two strands of DNA run opposite of directions is due to the positions of the carbon molecules on the deoxyribose molecule. Okay, and then again, the details of these things, I'm not too crazy about it, but just as long as you know it's anti-parallel, as long as you know it's a double helix, DNA is double helix. These are the things that I'm concerned about. Then the next one, since you know a little bit about the structure of DNA, the next one is how does DNA divide? One DNA becomes two. Okay, that's the next few slides coming up. This is called replication, copying, if you want. The original people, they like the term replication. That is fine. I call it copying. When DNA copies itself inside of a cell. I'm talking about inside of a cell, not in a machine. Okay, we'll talk about that it can happen in a machine too. It's called PCR. But inside of the cell, one DNA becomes two is called DNA replication. Okay, the process of copying of DNA. Okay, it occurs during the S phase of interphase. You learned that before, during mitosis and also meiosis. Each original strand serves as template. So when DNA is multiplying, one of these strands is a template. It means the other strand has to be built based on this one. That's what the template means. So this one is the original one, and then the other one is built next to it. Okay, that's what they call a template. Complementing new strands, DNA replication is semi-conservative. It means each new double helix has one original strand and one old one. What does that mean? If you have DNA like this, right? So this divides and become like this, right? So these are the old one, right? This is the old strand from here, and this is the old strand also. And then what happened, if I can, how do I get my colors changed? But anyhow, sorry about that. If they have the new one formed, the new one, this is the new one, right? These are the new one. That's what they call it, semi-conservative. It is not like scientists for a while, they thought, oh, this is the DNA. So you have the new one like that. Well, how? It doesn't happen like that. They separate, the two strands separate, and new one is formed by the old one. New one is formed by the old one. Let's move. Here they are. They're showing it to you. So this is the old one. If you look at it, this is the old one. This is the green is new one. The green is the new one. That's what it's called, semi-conservative replication. Okay, replication of DNA. The steps, these are the name of the molecules that are involved. Uh, you know, please study them. Make sure you know them a little bit. I'm not too crazy about it, but again, you got to study them. And I go over them quickly. If DNA is twisted like this before it multiplies, if DNA is twisted like this, you guys see that? Number one, DNA ligase unwind and unzip. So it becomes like that, right? It is twisted. DNA is twisted. Alpha helix is helix. And then it has to unzip, become like that, become straight, and then unzipped. I don't have a zipper with me. I should next time, I should get a zipper for next lecture. And then it separates, and then the new DNA polymerase adds a new DNA Sanas. Okay, sorry about that. I got disconnected. Let's move on and talk about this. My internet, I guess, disconnected me. It hasn't happened all semester long. It had to happen during this chapter. So anyhow, let's go over and hmm, oh, how do I? Uh, okay, let's see. Hmm. Okay, you guys, you can see me, but how do I? Um, oh boy. Okay, great. Now, okay, great. Now, I guess we can move on and talk about this. Um, so this protein molecule right here, it's going on zip and of course break apart. It's called helicase, DNA helicase, and it will break down the DNA from denature it. They call them, it's called it denaturation. They denature it. I'm not sure if I'm being recorded or not. Yes, recording. Okay. So right here. Okay, that's much, much better. And then these molecules, DNA polymerase, it comes along. DNA polymerase has two functions, and make sure you know those two functions. DNA polymerase, it comes along and it builds up the new, right here, it builds up the new, Luis Eugene, I'm not sure, Luis, why are you here? But I'm lecturing Louise. Anyhow, so it goes along and make up the new DNA molecule. DNA polymerase does it, and then also what does DNA polymerase do? It will go back and make sure the strands are matching. Okay, and that's another things DNA polymerase will do. Also, in this diagram, you see Okazaki fragments. They are little fragments here, and the protein molecule enzyme DNA ligase put these little fragments together, and this is the template. Remember, we talked about that template, and these are the new strands. Little by little, are being formed with each other. Okay, so replication of DNA, make sure you know the name of these enzymes. For mutation, if there is a mistake, for example, what happens in DNA, your intention is connection is again as unstable. Ah, but anyhow, A to T, G to C, and what happens, that is the original DNA. And what happens, there's a mistake. Here is A, here is T, right here, and here is a G, and here is a C. So the new one, it forms an A, right? The new one, that's a mistake, right? And then right here is a C, and this one is a G, and this one, there's no mistake here. These are mistakes, right? That's a mistake. That's a mistake. So what happens? The DNA polymerase does not know, does not have memory, and will go fix this one and put T in here. Or it will go fix this one and put G in here. This G to C and C to G is not same as the original one, A to T. And this is called mutation right here. That's a mutation. That's a mutation. So that's what mutation means. We've been talking about that all semester long, and now you're getting a hang of it, what that is. Okay, so having said all of that, again, I hope are you guys seeing my screen? Let me share. Let me. Oh, I'm not sure you're. Um, yeah, share screen. So okay, great. So Louise, you're here. That's fine. You don't have to be here. So I don't know. You guys caught the other DNA. I was talking about this when it got cut off, um, and then I hope it's all connected together. So here is a DNA helicase. So right here it unzip and break apart the DNA, and then DNA polymerase right here and DNA polymerase right here. It has two functions, same enzyme, same protein molecule. It has two functions. One, it makes the new DNA molecule. They call it Okazaki fragments. And then also it goes after it's all connected together by DNA ligase. I'm making the long story short, you guys. So the new strands, DNA polymerase goes and check the new strands, and then mutation can occur. When DNA polymerase is going back and checking, it does not have memory that it was A to T. If there is a mistake and a mistake occurs, it can change that one to C to G. Anyhow, so structure and function of the RNA. The next thing is RNA, and RNA stands for ribonucleic acid. The ribose molecule, it has the oxygen. So that's one of the big difference between RNA and DNA. Three differences you should know. RNA has the ribose has the oxygen. It's called ribose nucleic acid. RNA is single-stranded. DNA is double-stranded. RNA has uracil. So the RNA does not have thymine. Okay, instead it has uracil. So thymine is replaced with uracil. Complementary base pairing occurs. C pair with G and A pairs with U, usually single-stranded. So those are okay. This is, you guys, RNA and similarities and differences. Please make sure you study this and you know the differences between the DNA and RNA. So structure of RNA, and that would be the again, they have phosphate sugar, but the sugar is different. It's ribonucleic acid, and then nitrogenous base attached to sugar molecule. And then you do not have in DNA, in RNA, I'm sorry, in RNA, you do not have thymine. Instead, you have uracil, you have adenine, you have cytosine, you have guanine, you have all of those except your regular normal thymine does not have it. Okay. There are different types of RNA that you should know. Types of RNA are messenger RNA, ribosomal RNA, transfer RNA, and small RNAs. They are the big bulk of what you're supposed to know are these and that one, which I'm going to elaborate on it. Not much. I'm not going to elaborate. Your textbook has elaborate on small RNAs, which is okay, but your course is not designed to focus on those, and I will talk about it. Messenger RNA or mRNA, when DNA separates, and now I'm getting into how a molecule, protein molecules are going to be synthesized based on DNA. That's the next step of this chapter. So the messenger RNA, as the name applies, it gets the message from the template, from DNA, and it goes into the cytoplasm and starts putting the amino acids together to make protein molecule. Okay, so produces in the nucleus where DNA serves as a template, carries genetic information from DNA to ribosomes in cytoplasm, which we talked about that. rRNA, the function of it is still fuzzy, but it monitors the whole thing. It's a RNA molecule that goes into the ribosomes and is inside after it's being synthesized in the nucleolus. It goes to the ribosome, and then in the ribosome, they monitor the whole thing. Ribosome is made up of large and small subunits, protein molecules. You have the large subunit and the small subunit protein molecules like this, and that's what ribosome is. They leave the nucleus and then attach to the endoplasmic reticulum of the ribosome cytoplasm. During protein synthesis, they combine from ribosome. Okay. Transfer RNA, that's another important thing you should know. It produces in nucleus using DNA template again, and they are the one that attach to amino acids. They are the only DNA RNA molecule. They are the only RNA molecule that attach to amino acids. Okay, so transfer amino acids to the ribosomes during protein synthesis. Make sure this is a quiz question. There are only, we human have only 20 different amino acids. Chapter 2 or 3 did not list them all, but you should know we only have 20 amino acids. That's a number. You know, usually in these classes, I do not expect you guys know numbers, but yes, this time you should know numbers. Like we have 24, 26 letters in the alphabet. My God, look at me. But anyhow, we have only 20 amino acids. With those 20 amino acids, you can have billions and trillions of different people on planet Earth. Anyhow, each type of tRNA carries a specific amino acid. There are 20 different tRNAs. So small RNA divide into several classes. So small nuclear RNA, snRNA, involves in splicing the mRNA before it is exported from the nucleus, which I will talk about that. Small nuclear nucleolar RNA, SON, are attached to mRNA in the cytoplasm, prevent unnecessary translations. And I will talk about what translation is. Hang on. Small interfering RNA, siRNA, also binds to mRNA and prepares the mRNA for degradation. Break. At the end, when everything is done, the mRNA needs to be break apart. Imagine this would be DNA and this would be mRNA. mRNA is being synthesized based on one of the strand of DNA is stranded like this, and then it leaves the nucleus and goes into the cytoplasm and continue the protein synthesis. Okay, so gene expression, that's the next title in your chapter. It means based on this DNA, I am short. My hair color is dark. My skin color is brown, whatever it is. My eye color is blue. It's not blue, you guys, you know that. But that's based on this. This decides, this DNA decides I'm blue color. Okay, let's go. That's what's called gene expression. My earlobe, last chapter, is not attached. I can roll my tongue. Okay. Gene expression, DNA acts as a template for making RNA, which in turns act as a template for manufacturing protein synthesis. During gene expression, protein synthesis, transcription, it means RNA synthesis. This is DNA. When you make RNA, this is RNA, you guys. When you make RNA based on DNA, that's called transcription. When you make RNA, this is RNA, when amino acids, imagine these are, I wrote down AA, you guys can see that, AA, my abbreviation, Amir assadi-rad. No, it's amino acid, but anyhow, this is mRNA, this black pen, and then these are amino acid, that's called translation. Okay, transcription, translation. I hope I'm making some sense. When proteins, of course, Proteins are made up of amino acids. So 20, again, your textbook is mentioning there are 20 different amino acids, quiz questions. Proteins differ in numbers and orders, and amino acids, the sequence of amino acids, protein leads to a particular shape and so on. Transcription, he's saying it, the first step of gene expression, that's the first step. First step, you must, the cell, bacteria, elephant, whale, flowers, fungi, first they do transcription. And then they do translation. Okay. So transcription is the first of gene expression, the sequence of nucleotides in DNA copied, sequence of nucleotides in mRNA, transcript, and then translation. The second step, mRNA is translated into sequence of amino acids. That's translation. I hope I'm making some sense. Right here, they're showing it to you. So if this is the DNA, this is the DNA, that's a template. So A, T, right? This is mRNA, right? Right here. mRNA, GC, CG, CG, TA, GC, GC. Oh, they don't have U. Son of a gun. Wow. What if you had A here? You should know these things. A, U, uracil. You guys follow what I'm talking about? Again, your textbook made it a little bit of a mistake. And then based on these three nitrogenous bases, these three are responsible for amino acids. These three are responsible for an amino acid. These three are responsible for amino acids. And then these three as a stop signal. But anyhow, let's say that's okay. Genetic code based on DNA and mRNA codes for amino acids, codon, three nitrogenous bases I mentioned, and there are 64 different possible codons. Yes, I would like you to know that. And the stop codons, there are three of them, and start codon is AUG, like month of August, when school starts, usually in August. Most United States, school starts in month of August. Okay, right here. That's the codon to start a protein synthesis, universal, all over. Here it is, 64. When I say there are 64 different codons, and right here, it would be interesting that you would know this, but again, that is, I think it's beyond the focus of this class. I will not ask you any question, but when you go to the next class, they might ask you to know this table. Do not memorize it. By all means, do not memorize this table. But maybe for next class, you should know how to use it. If you're going that route, you're going to biological sciences. Most amino acids have more than one codon. Yes, that's what it means. Look at the amino acid histidine, for example, right here. CUACAC. Glutamine, CCA and CAG. Okay, so serine, amino acid, these are all amino acids. Serine here, there should be serine here. So it is possible to have, that's why you have 64 codon, but you only have 20 amino acids. So for one amino acids, you have several codons, depends. Some 2, some 4, you saw in case of serine. And then you have 3 codons to stop at the end of the, right here, at the end, it's a stop signal, say stop. No more amino acids needs to be attached. Okay, there's supposed to be a third one. Where is the third one? UUA, UGA, and UUU. Where is it? But I don't know. Anyhow. Anyhow, there are 3 stop codon. Genetic code, most amino acids have more than one codon, and this redundancy is in genetic offers protection against possibility of harmful mutations. That's right. During transcription, a segment of DNA serves as a template for protection of RNA. All classes of RNA are formed for transcription. RNA polymerase enzyme that adds RNA nucleotides using a complementary base pair rules, and then law of base pairing, as I said, A in DNA is A to T, G to C. Of course, when it comes to RNA, A to U, T to A, G to C, C to G. Make sure you know these things. That's called law of base pairing. So here they're talking about it, and then what happens when a piece of mRNA is synthesized, mRNA. When a piece of mRNA is synthesized, what happens? Segments of DNA that interrupt genes and are not part of the gene, they do not code for functional proteins. These are called introns, right here. Okay, for example. And then the one that are going to get translated and they go to cytoplasm. This is all mRNA synthesized in nucleus, right? And then portion of it, not all of it, are going to go to cytoplasm. Those portions right here, right here, right here, and right here are called exon. They will be exit out of nucleus. They go to cytoplasm and they get translated. Okay. Here they are. They're showing it to you what is exon and introns and so on and so forth. I'll let you study those. During processing, one of the end of the RNA capped. Again, go on in nucleotides. I'm not going to go over those details for your class. If you take Bio 1, I will talk about it. So this process, the mRNA go through process of being chopped off and pieces being put together, right? That's called splicing. Just like when you go to the kitchen and you splice things. But in the kitchen, you cannot put them back together. Well, then when you put them in the dish and then you eat them, yeah, I guess that's, but here they're physically attached. Those exons will attach to each other, the exons. So there is a protein molecule called spliceosome, and that puts the exons together. And there is an enzyme. This is a quiz question called ribozyme. Ribozyme is an enzyme. Before ribozyme was discovered, textbooks wrote down all enzymes are made up of protein. And here we go. They found an enzyme that is not made up of protein molecule. It's made up of RNA. So this is the only enzyme on the planet Earth, so far we human beings know, that is not protein. It's a RNA molecule. Okay. Alternate RNA, some cells, only certain exons and genes results in different protein molecules. Small RNA regulates not only mRNA processing, but also transcription and translation. Translation complex. So the next step after transcription, and you learned about splicing and so on and so forth. Now we are getting into the cytoplasm, and in the cytoplasm of the cell, translation is occurring. Transcription occurs in nucleus. Translation occurs in cytoplasm. So the RNA brings amino acids to the ribosomes. And you remember a ribosome, you have a large unit and small unit. And translation complex is small and large ribosomal subunits together bound mRNA. And the ribosomes contain binding sites called APE sites. And I will go over that, where individual tRNAs attached to. Here is a protein molecule, amino acid. All of these blue balls are one amino acids, and they are being put together. Here's an mRNA ribosome. For example, this is mRNA ribosome. So it comes through and it's going through. tRNA comes in, drops off the amino acid and goes away. This moves. tRNA comes in, drops off amino acid. It it goes away. This moves, tRNA comes in, drops off amino acid, goes. tRNA comes in, drops off amino acids, and goes. That's how it works. Okay, guys, tRNA, it came from where? mRNA came from where? From DNA, goes to ribosome. tRNA, attach amino acid to it, comes off, drop off the amino acid, and go away. One amino acid, one tRNA, one amino acid. Okay. Right here, they're showing you a tRNA molecule right here, and tRNA, the 3 nitrogenous bases on tRNA is called anticodon. On mRNA is called what? Codon. Oh, the A site, this diagram is showing you the A site where it arrives, okay? And then P sites where the tRNA drops off the amino acids right here. And E sites is exit sites. After they drop it off, the tRNA goes here, and then after that, it leaves the ribosome, and the next tRNA comes in. Okay. So the tRNA has areas of binding amino acids. It's called anticodon. I showed it to you in this diagram. So right here, for example, if the mRNA, the mRNA is UCA, what would be tRNA? You should know this. Okay, tRNA, we do not have T, A, right? U binds G, CG, and A, it would be U, right? That would be, if this is mRNA, that would be tRNA. Let's go. The anticodon has 3 sequences, complementary base pairs, codon, mRNA. Each amino acid has a tRNA that carries it to translation, and so on and so forth. So here it is. Here is amino acid, and here is anticodon CUG. It could be anything. Remember that. It depends on what is the mRNA. Translation concluded. So there are the order in which tRNA links to the ribosome is directed by the sequence of mRNA codons. The tRNA attaches to growing polypeptide now, moves from ribosome to A sites and P sites. Okay. So translation has 3 steps. Make sure you know these things: initiation, elongation, and termination. Initiation, mRNA binds to the small ribosomal subunits. This initiates the formation of translation complex. And here they are. They're showing you the A sites and P sites and E sites, exit sites, arrival sites, and so on and so forth, which elongation, polypeptide length, amino acids, so on and so forth. When amino acids are attaching here, first amino acid is methionine. Make sure you know that. Month of August. Remember the the codon is AUG right here. That would be the codon. The codon is always AUG. Protein synthesis always starts with AUG. Okay, and then the first amino acid is methionine. Okay, so what am I saying? Elongation, when amino acids are attached here, another amino acid, this is called elongation. So elongation, polypeptide, I mean, poly means many, peptide, it means the amino acids. And incoming tRNA arrives at the A sites, the ribosome moves down the RNA codon. As I said, it moves, the mRNA must keep moving. I hope you all know what the spent tRNA now, the E sites exit the ribosome, and so on and so forth. The A site is now available to receive another incoming tRNA right here. Just showing it to you again. Termination, there's supposed to be a stop codon on the mRNA at the termination sites, the very last codon, stop codon, which they're talking about it here. Okay. Polyribosomes, multiple ribosomes moving along one mRNA. It is possible at the same time. Again, make sure you understand these things, you study them a little bit. And if you have any question, please ask. This is a summary of what's happening. Here's a nucleus right here. So transcription occurs, mRNA comes out, it goes through the ribosome, possibly more than one ribosomes, two, three ribosomes, and tRNA comes in, drop off amino acids, and polypeptide are being made. And then tRNA keeps coming and drop off. Here is a tRNA without any amino acids. Here is a tRNA with amino acids, and they drop them off. And then pretty soon you have a protein molecule. Here they're talking about some of the molecules participant in gene expression. Some of it, they should say there's a lot more than this. But be familiar with them. It's not much. It's very simple, straightforward. Then everything has a regulation. We have a society, there are regulations in the cells, there are regulatory. You've studied it all along, mitosis is being regulated. So the protein synthesis is also being regulated. You have a thermostat at home that is, it regulates the temperature inside of your home. So for regulation of gene expression also exists. Levels. I have it in, you have RNA being synthesized when it comes to the cytoplasm, protein synthesis, all of the different levels of regulations as far as making new protein molecules. Here we go. They're talking about it, access to its gene also is involved with DNA. So anyhow, levels of control, they're summarizing it here for you guys. Very good. Okay, so they're at all levels. Transcriptional control, several mechanisms regulate which genes are transcribed at the rate which their transcriptions and so on and so forth occurs. Post-transcriptional, yes, occurs, I think I asked questions on this one, occurs in the nucleus after DNA is transcribed and the mRNA is formed. More regulations, yes, please study them. I'm not going to go to them in great detail. It is kind of important. You should know that. Transcriptional factors, DNA binding proteins after transcription factors binds to DNA, RNA polymerase attaches to DNA. Well, they're protein molecules that are specialization in determining which genes are activated or not. Okay, let's talk about some technology, DNA technology, which in last, I would say 20, 30 years, oh my God, a geneticist came on the radio, said, the things we are doing nowadays, at the beginning of my life, when I started getting into the field of genetics, we dreamt of these things that we are doing now. So it's the field of genetics is exploding so much that the laws of ethics cannot keep up with them. It's amazing what they are doing. Things that they are doing. When I started graduate school, I never thought it will happen. Just fixing genes with CRISPR gene. I would think that it would happen 100 years from now, but it's happening right now. It's amazing how the field is moving forward with the technology, DNA technology. So anyhow, understand the goal and editing. So let me get into DNA sequencing. So one of the things, the person, usually people who win the Nobel Prize, they usually are 2, 3 guys from different parts of the world. They win a Nobel Prize. This guy at Berkeley, UC Berkeley, right at the back of our, he won the Nobel Prize all by himself because he came up with polymerase chain reaction, PC You probably have heard about it, PCR, okay, on the radio, TV, PCR. He is the one at UC Berkeley discovered it, so what he did, he put a piece of DNA into a machine, and then that piece of DNA gave you 2, that 2 gave you 4, that 4 gave you 8, 8, 16. You keep repeating it, you have millions of copies in a matter of hours. So these are the steps of PCR denaturation. It means the DNA has to be broken in half. For example, you find a piece of DNA from a dinosaur. You find a piece of DNA from a crime scene. Okay, you find a piece of DNA in mummies, and you want to make copies of them, many copies of them, and sell them to people. For example, I'm just saying some stuff that makes sense. Then, right here, you put it in a machine, million copy at the end. So the first step is to break that DNA, denature it. The next step, and then they add chemicals in here. So 2 new strands are formed now. DNA polymerase added complementary bases to the new strands, creating 2 double-stranded DNA. That's called extension. And then you're repeating that, you repeat that. At the beginning, when you heat the DNA, it breaks it up. And then if you cannot keep heating it forever, you have to cool it down. And the guy who invented, he came up with a machine that he heated up, and then he came manually cool it off. And then he added a polymerase codes and so on and so forth. Then he had 2 strands. Then he went heated up again, DNA broke apart, and then he cooled it off, and then the primers, everything was added, and then he had 8 copies. Nowadays, it's automatic. The machine will heat up itself up and it cools itself up. Okay, right here is telling you what's happening. Please study it on your own. DNA fingerprinting. So we are all made up of different DNA, and our DNA sequence is different. So with the help of PCR, you can multiply it. And if you have 2 suspects, suspect A and suspect B, and then you collect some DNA from crime scene, and then you pass them, and then you can identify, suspect A is, that's what we call a DNA fingerprinting. Okay, it's more accurate than your fingerprints. Okay, of course, there are mistakes can be made, there are things can happen, but you can run it again and you can do it in different laboratories and they give you hopefully a better results. Cloning, it means you take one piece of DNA and make a copy of it. That's called cloning. Or you take an animal and you copy it, is called cloning. So anything, piece of DNA, a cell or organism, it can be done. Recombinant DNA is called rDNA. It contains DNA for more than one source, and use a vector. Usually vectors are viruses. They can transfer DNA from one thing, one organism, or from one bacteria to another bacteria is called vector. Plasmid are circular DNA, which is found in bacteria. And then we use it to insert a DNA in here and get the right products. They are showing it here. They take a DNA that we want to make, for example, insulin, and they take the plasmid out. They put the insulin gene here, right? That's the gene that gives insulin. They put it inside bacteria, and that bacteria gives you the insulin or growth hormone. Whatever protein molecule you want, the bacteria make it. Okay. Cloning, steps of gene cloning, restriction enzymes. Yes, restriction enzymes are enzymes that can cut off DNA, and they're called restriction enzymes. Again, they get them from bacteria. Restriction enzymes are present in bacteria, and DNA ligase, again, they get it from bacteria and put the genes together. So when you break the DNA with help of restriction enzymes, you have sticky ends. They call it sticky ends. See, that T binds with A, and T binds with A, and A binds with T, and T binds with A. So they call this sticky end. When you want to insert another DNA in here, so that DNA must have A, T, A, and they can stick in here, the gene for human, okay? The gene that makes insulin or growth hormone for human. So this is from bacteria, okay? Let's go back. I think this is from bacteria, right? This is sticky end here, sticky end here. They get a gene from human right here. That gene, they want to put it in here. That must have sticky end here, sticky end here on both the desired gene and the plasmid. Anyhow, steps of gene cloning. Here we go, study them, please, if you know. Reverse transcriptase, it means it's an enzyme that can make DNA based on RNA. That's what they call it, reverse transcriptase. Like HIV has that enzyme, reverse transcriptase, which HIV does not have DNA, it has RNA. Based on this RNA, they go inside of the cell of white blood cells of human, and then based on this DNA, they have the enzyme reverse transcriptase. They can make DNA. Based RNA, they can make DNA. Reverse of transcription, where the enzyme ACE at the end of the word is an enzyme, that is possible. So anyhow, complementary reverse transcriptase, they make complementary copies of RNA into DNA, and then genomic editing. CRISPR gene. So earlier, it was just recently discovered with this, the CRISPR, they can go into my defected cells of my body, and they genetically defected cells, and they correct those genes. That's astonishing. Nobody thought of we'll be able to do it 20 years ago if you would have talked to any geneticist. But it is possible with these CRISPR genes, they call it. They can go ahead and fix. I'm not going to go to details of it, but it would be interesting right here. So with PAM sequencing, they can go ahead and, of course, there are a few other things that are involved. This is the original DNA. I'm sorry, I'll break that up. Double-stranded, they can't fix the gene of a person that is defective. Biotechnology, genetically engineered. So you've heard of GMO, genetically modified organisms. When you go to the store, GMO in the United States, it did not pass through that the food that we eat, the labels of the food we eat, it has a GMO labeling on it. In Europe, it passed. So when you buy a food, you can read the label. Yes, it has a GMO, not in the United States. Good or bad, I don't know. I don't want to get into politics. But when the genes of an organism, for example, they make some kind of fruit, apple, that withstand frost, or oranges, that in Florida, when the frost come, it does not affect the oranges. Okay, so that's what genetically modified means, GMO. So there are, here we go, the apples. This is the natural apple. Here's a GMO apple that when they are exposed into the air, they do not turn color. Okay, they can do that. People do not like an apple to turn brown. It's nothing. It's no big deal if it turns brown. Believe it, guys. Talk to a people in the nutrition. If it turns brown, there's nothing wrong with it. but people don't like it, so they go into GMO and they make a GMO soybean. And then the examples are a lot in here. From animals, they make animals that they make more wool or other things. Right here, they keep talking about these things. Gene farming, okay, right here. If you want to make growth hormone, human growth hormone, for example, right here, you take the gene. Before they used to do it with bacteria, now they can do it with mammal cells. You take the gene and you put it in a goat's nucleus cell, and then you can do cloning. Your chapter didn't go into cloning very much, but that's okay. So with cloning the animal, you got an animal that can make human growth hormone. So you can clone that animal right here, and then you collect a lot of human growth hormone. Other things that is possible is xenotransplantation, which is a transplantation of animal organs or tissues into human. So again, if we do not want to reject the heart or kidney or liver of an animal, we have to genetically modify it to our genes. So my body does not recognize that as a foreign molecule and be able to put it inside of my body. Okay, I think, genomics, study of genes, and then human genome. This is a quiz question. In 2003, a complete human genome was discovered. So believe it or not, a lot of people were surprised. There's only 3,000 genes we have, which is useful. You're talking about useful genes. We have, anyhow, we human have. And the person who is next to you, 99.99% similar to you, has those 3,000 genes. Okay, that's pretty much what I'm talking about. Genome sums, productions, and so on and so forth. 2% of human genomes codes for functional proteins. Out of those, and 98% remain produce molecules that regulates genes. Okay, I mean, I'm not gonna go into details of that. How our genes function has identified rules of intergenetics and so on and so on. Comparative genomics, comparing the human genome to that of animals, and then sequence of chimpanzees and human is 95 to 98%. We are similar to chimpanzees. 85% similar to mouse. Yeah, I don't know. I'm not going to ask. Proteomics, like genomics, proteomics is the study of protein, human proteome, the proteins of human. Mainly, it's study of human proteins and comparing and three-dimensional, a lot of things of human. Bioinformatics using computer to study the genomes with help of computers. Gene therapy, again, insertion of genetic materials into, we talked about that, the CRISPR gene. Both ex vivo outside the body and in vivo inside the body gene therapy methods are being used. Ex vivo gene therapy outside of the body, please read it, and then your textbook says that it can be fixed. Combined immunodeficiency disease, SID, hemophilia, Alzheimer. It's not there yet, but they're hopeful. Scientists are hopeful. And here they are, the gene therapy, they're hoping that it can happen. In vivo gene therapy, it's a little bit different than they use viruses as a vector. We talked about that to fix some of the genetic disorders. All right, guys, I hope this whole entire thing worked.